Thank you for the invitation. It's great to be here. I'm in the School of Biosciences, and my specialty is cancer research and cancer therapeutics. And I know I'm not meant to be giving a career talk, but I thought I'd try and put in some career things as a bit at the beginning and a bit at the end about careers. And, we all draw our careers like this or some nice line, although it was very interesting to hear Sharon's. But to be truthful, everybody career looks a bit like this. So, okay, I'm a professor of cancer therapeutics and whatever. But, I started off as I did A levels and I was undergraduate at Leeds and didn't get the degree that I wanted quite. I got a 2-1, not a first. And and then I went and did a PhD at the Institute of Cancer Research in London. And after that, I really wanted to go and study research in another environment. And so I thought, well, why not try America? So I wrote to lots of people there, and actually some of them gave me interviews. I didn't look for a job advert. I just wrote to people that I was interested in. And I found someone at Yale who was working, and I'd originally worked in cancer for my PhD and on signaling of this then hot new oncogene in the 1980s called RAS. But I decided for my postdoc to go and work in a different model system, but on the same types of proteins that were related to RAS. And I was very well. It took me, I think, about 5 goes to get a fellowship, maybe 6. So I definitely say to you all, if at first you don't succeed, keep going. It's like with grant writing, there's lots of failure. But eventually I won a very nice fellowship to go there. But it wasn't plain sailing. And the reason why there's this dip here is because I realized that yeast wasn't for me and that I really wanted to go back to cancer research. So, but I stayed and learned a lot of molecular biology techniques because I thought I don't want to jump ship. I've got this fellowship, for 3 years. So I stayed and my supervisor was lovely and I got some nice publications out, but I spent a lot of time thinking how I was going to move back into cancer research. And I thought about doing a second postdoc, but eventually through networking and going to conferences, I ended up working, interviewing a company called Onyx Pharmaceuticals that was a biotech company based in San Francisco. And this was actually through the person that ran that company was someone that I'd actually knew from my PhD. So, networking is very important, is all I can say. I stayed there for 5 years, learned a lot about how to do cancer drug discovery, had a collaboration with Pfizer. This led eventually to a drug called palbociclib in breast cancer. And it targets the cyclin-dependent kinase 4, which is involved in the cell cycle. But eventually I realized that I wanted to come back to the UK. And actually, again, through talking to people in the UK and actually talking to my former PhD supervisor and other people, I realized that there were drug discovery jobs going back at where I did my PhD. It was sort of a circular thing. And but I went to another division to do with cancer therapeutics and was there for 15 years and was in this Cancer Research UK Cancer Therapeutics unit. And it was fantastic. And I learned a lot about drug discovery and interacted with lots of companies, including AstraZeneca, which I'm going to talk about. And then in 2014, I moved to the University of Kent, where I still do drug discovery, but it gave me also the opportunity, and you don't fall off your chairs, to actually teach. Because it was something, I'd spent a lot of my career doing drug discovery, but you feel at some point that you want to give back. And I really, I teach biomedical science. I teach to the medical school here. So, and I still do drug discovery. And the project that I'm going to talk about today is about the AKT inhibitor that actually James talked about. I'm going to talk about the origins of the project and how it got to approval. And I guess, just to say that there's, your career will be, I'm sure, very long and there'll be ups and downs, but just stick with it. All right. I shall now move on that note. So I've been working in cancer research for the now I'm starting to get really worried about this. It's about 35 odd years. And you know, we know now that 1 in 2 people born after 1960 in the UK will be diagnosed with some form of cancer during their lifetime. But the most important thing to realize, you know, it is a very, when you first see that number, it's very scary. But, you know, a lot of what we've talked about today and what's going on in cancer research, we're changing 10-year survival. This is actually probably out of date, but it was the only data that I could get hold of. But survival is improving in most cancers, and it's due to faster diagnosis and actually due to, you know, drug discovery. So treatment advances. And with prostate cancer, we've seen the largest improvement over this time span. So from 25% to 84%, and it's even higher now in 2024. And I've worked in both prostate cancer and I'm very interested in breast cancer, and that's undergone a huge change since the 70s. And actually that is now up at, it's over 80%, 90% now in terms of 10-year survival. So, you know, it's people like us and, you know, you who are making these differences. It's incremental, but it's very important over time. And I'm probably preaching to the converted here because I know you've done your translational module and everything. But I always like to show this and just, you know, emphasize the point that drug discovery really is a multidisciplinary activity. And it involves lots of different types of scientists, you know, whether you're interested in cell or molecular biology, whether you end up being involved in computational biology, pharmacology. And we've heard about the in vivo biologists in particular today with James. Chemists and the different types of chemists that there are. I spent a lot of time with medicinal chemists, but also I spend time with physicists to do with imaging and clinicians as well. And it's all those different types of people at different stages of their career that make up, you know, a successful drug discovery project. And it's going all the way, and academics. And I've been an academic and in industry, and all the way from target validation, so identifying a protein or gene that's altered in cancer, all the way through the drug discovery pathway that you've seen in various forms, identifying compounds, optimizing them, doing the in vivo biology experiments, and then going into clinical trial with drugs and hopefully approval. And for me, one thing that's become very clear in what I do, and mainly what I've done is been involved as a cell and molecular biologist all the way from target selection and actually all the way into the phase 1 clinical trials. For me, it's biomarkers, because they've been very key in driving the drug discovery projects. And you've heard sort of examples about it today. And the two types that have been specifically key for me have been predictive ones. So that's how we can identify the patients who are actually going to respond to the drugs, because you don't want to be giving drugs to patients that aren't going to get benefit from them. And pharmacodynamic biomarkers, so which is basically what the drug does to the body and effects that you can see on the body. And I'll give you some examples of that. So there's lots of people involved, and really one of the key things in drug discovery is how to make a good drug. And as I said, you know, we have to start with a drug target, but I think it's also the relationship between the different scientists that's really important. And especially when you're actually doing a drug discovery project, I found as a biologist that one of the key critical things is the interaction with the chemists, because they're designing the molecules. And the mission for the chemist, and my arrows seem to have moved, is how to put all the required properties that you need for that one molecule that's going to target a mutated protein in a tumor into one molecule. You know, it has to be able to be selective for that protein. It has to be able to travel around the body, perhaps to the tumor, to have good PK pharmacological properties. And, you know, how's the chemist going to make that molecule? Well, they don't make it in one go, that's for sure. They have to make hundreds of them. And for the biologist, the mission is how to identify that one molecule during the drug, you know, and so we do it in iterations with all these multiple optimized properties. And so designing the experiments and the assays in particular is really critical for people like me involved in drug discovery. but it won't work if we don't communicate. So communication's very key in drug discovery. As I said, it's team science. So I'm going to give you an example of that. I'm glad that Steve finished early because I'll probably hopefully I won't run over too much, but I'll speed up. So I think somebody else, was it Lucy, introduced, couldn't be there for all the talks, but the protein that I'm interested, that I'm going to talk about and that we developed a drug for, along with the, it was AstraZeneca as well, is AKT. And AKT is a kinase that sits on this pathway known as the PI3 signaling pathway. And it starts with signals from outside the cell that go through growth factor receptors that activate PI3 kinase and eventually AKT. And then AKT can phosphorylate other proteins that are involved in driving cell proliferation, cell growth, and survival. So you can imagine if you get a mutation in any of these things, it's not going to be good because you could be driving inappropriate cell proliferation or survival. And as you may know, these are key hallmarks or physiological characteristics of cancer, uncontrolled proliferation and survival of that cell when it shouldn't be. It should have undergone cell death. And so it's on an important intracellular signaling pathway, and these just stars highlight the proteins that and their genes that are mutated in cancer. And this table just shows some of examples of this, of the percentage of mutations of just AKT itself in different forms of cancer. You might call that part of the target validation, why we'd want to target it with a drug. Which takes me on to the next slide. So when we think about, are we going to do a drug discovery project? And a number of people mentioned this, we have to think about various things, because you do not want to initiate a project unless you're absolutely sure it's the right thing to target, because it's going to cost millions, all the way from, identifying the first compound, making the best compound, putting it into clinical trial. So you've got to be pretty sure that it's a good drug target. And part of it is, yes, this thing about, is it mutated in different forms of cancer? And this is what this relates to. But it's also other things that are really important. And again, I'm probably preaching to the converted if you've done translational science, because you've probably heard words such as druggability, as in, can you design, does it have a cleft where you can design a molecule that can bind to? The reality is, if you've got flat surfaces interacting together, it's very hard to design a molecule that will bind to a flat surface. Pockets are much better. Can you take a structure-based approach? Is there a crystal structure for your protein? That really helped us on AKT, and I'll come back to it. Really mundane things like, can you make large amounts of protein, of your recombinant protein in bacteria so that you can design biochemical assays to screen compounds? If you can't do that, it's going to be very difficult to execute drug discovery project. And again, can you, if you make compounds, how are you going to see whether they're hitting your target? Are they inhibiting AKT? Well, we could look at phosphorylation of these downstream substrates using antibodies that detect the phosphorylated versions of these proteins. And luckily for us, there were antibodies available for us to do that. And also this thing about, how do we select the patients? Well, if we know these things are mutated in the tumors, one hypothesis is that those patients who have these things mutated, such as AKT or the phosphatase P10 or PI3 kinase, should be the patients that we would want to target, because their tumors should be more sensitive to these drugs. Right. So I've told you a bit about, what you need to think about when you're going to propose something as a drug target to somebody who's got a lot of money and is willing to fund you. In this case, it was Cancer Research UK, and it was while I was at the Institute of Cancer Research and then more recently at Kent. And so back in, probably, I hate to say this, before some of you were born, back in 2000, when I was at the ICR and a junior group leader, in our site visit, I proposed AKT as a drug target, because of some of the things that I've told you. And because it was a pathway that I was interested in. And they decided it was a good drug target and allocated funding towards it, but we really couldn't start the project for a while because we didn't have the structure at the time This came along a little bit later from this individual. And there was some technology that we needed. And we wanted to do a type of screening called virtual screening, and we didn't have it at the ICR. But if you don't have something, find someone to collaborate with. And so again, through networking, we spent time talking to Aztecs Pharmaceuticals, who are a company based in Cambridge. And I know that one of your apprentices, and I can't remember her name now, who worked with Nicola Wilshire at Aztecs. I know that one of the apprentices who was at Kent was based at Aztecs Pharmaceuticals. So they had a technology that we were interested in. So we went to talk to them and propose AKT as a drug target, and they thought it was a really good idea as well. So we joined forces together. So it was sort of Aztecs, Cancer Research UK, who were funding people at the Institute of Cancer Research, including me. And these are some of the key people that were involved. I was the lead biologist for the project, and there was a chemist, and I was the overall project lead for the ICR. And then I had colleagues at Aztecs, a lead biologist and a lead chemist. And also we spent a lot of time talking to a key clinician who was going to run the phase 1 clinical trial, and also our pharmacologist. So again, it's an example, if it's not one person working on their own, but you'll realize it's literally hundreds or thousands on a project. But these are some of the key players. So I'm watching the time, I'll speed up. So with Aztecs' technology, which involved also an individual called Gordon Saxty at Aztecs, and also the fact that David Barford solved the crystal structure of AKT at the ICR, which was really important, because what it allowed us to do was to look at that structure in silico and then do virtual screening of small molecules. And basically, you could say it was like taking those molecules and these small molecule fragments and testing their ability to bind virtually into a computer model of AKT. And you could say it was computer says yes or no, if you get my drift. But we got a number of molecules that seemed to fit into the ATP binding pocket, because it's a kinase of AKT. They were very weak binders in terms of their ability to inhibit AKT, but that's because they were small, so they weren't binding very strongly. But we identified 15 fragments that bound in virtually, and then we confirmed that using crystallography, so actually soaking these fragments into AKT crystals in the end, to confirm that they actually did physically bind. So virtual and then physical confirmation. And one of these we called series 3, and it's shown here. And it's known as, hopefully if there's any chemists on there, they won't shoot me. It's a 7-aza indole, if I've got this right. And it's a very small molecule, but what it allows you to do is add things on. And sometimes the chemists call that decorating them, or synthetic flexibility, that you can stick different bits on. And that means that it will bind better into the pocket and have a higher affinity for your target. We actually did something called scaffold hopping, which is we changed the primary scaffold here. If you look, this one's got more nitrogens on it, and this meant it was more synthetically tractable, which just means that medicinal chemistry wise, it was easier to add things on. And that's what the medicinal chemists did, and some of them are shown here. I don't know whether Steve's still on, but if he is, he knows all of these chemists. And so they added a basic amine for additional binding into the ribose pocket down here. And this increased the affinity and therefore lowered the IC50 towards AKT. So this is the IC50 for AKT. It started off as being a very weak inhibitor, now much stronger with sub-micromolar. That makes us, the biologists, happy. And then eventually through iterative chemical synthesis, crystallography, biochemical assays, and eventually going into cells, we ended up with this lead. By adding this lipophilic group in, which binds into the P-loop region of AKT and provided, you could say, more hooks into the ATP binding pocket. And it became a very potent inhibitor of AKT. And we ended up with this molecule, which I'll just call, it was our initial first lead, 930. And as well as managing the project, my laboratory role was to develop all the cell-based assays and look at mechanism of action and prove that it was inhibiting AKT. And some of the work was actually done by a very talented clinical fellow. So this is a clinician who's decided to do a PhD, and he's shown here. And he looked to confirm that the inhibitor was actually blocking AKT activity. So this was a human glioblastoma cell line where we do a 1-hour dose and of titration of the compound up from 0.1 micromolar up to 60. And we could see that by Western blot, we were inhibiting phosphorylation of downstream targets. So we were pretty sure that we were inhibiting AKT in cells. And then we went in vivo. As James mentioned, this is really important in understanding scheduling, which I'm not going to really talk about because I'll run out of time. But what we saw is that when we did pharmacodynamic assays initially, where we looked at 6 hours, which is very close to what James said at 4 hours for Capivasertib, we could see, again, when we took the tumours out of the mice, that we could see inhibition of the downstream target. And we could do that in a few mice and then eventually do more mice with an efficacy study in a breast tumour model that's shown here, and we could see inhibition of tumour growth. And this is just showing some of the individuals that were involved in these studies. Tim, who was a clinical fellow doing a PhD, Lisa, who was a technician, and Sue Eccles, who ran the in vivo group. Eventually, through lots of chemistry, that compound we discovered with our pharmacologist flow was not very orally bioavailable, which means that if you gave it orally to mice, it didn't really get into the bloodstream and around to the tumour very well. So eventually we progressed, we did more chemistry, or the chemist did, and we did more cell-based assays, and eventually found a compound that we could give orally to mice. And we could show again that it inhibited growth of the tumour in the mice. And we also could, by looking at phosphorylation of a downstream target of AKT in the tumours, show that we had inhibition. So it was very important to join up the efficacy with actually showing that the target was being inhibited in the tumour cells. We call that the pharmacological audit trail. However, you're never doing anything in isolation, and AstraZeneca also had an AKT inhibitor project, as did Merck, plus Pfizer, plus lots of companies and lots of other academics. And AstraZeneca came and we're always talking to each other. This thing about we're not talking to each other is utter rubbish. We're always talking to each other. And eventually, I think it was Donald Ogilvy at AstraZeneca, along with Martin Pass, who came and had a conversation with us at the ICR and said, oh, we're running an AKT inhibitor program, but we like your molecule. Would you be willing to let us evaluate it in comparison to some of ours? And we said, we eventually knew that we couldn't, it would cost a lot of money to put this into a clinical trial, and we were going to have to partner with a large pharma company at some point. So we said, yes. We had a big debate internally. So that was CR UK, Astex, and ICR, and we thought this is good, and we like AstraZeneca, and we think they're a good person to partner with. And so in the end, we licensed this compound to AstraZeneca, and they did more chemistry on it. They put an additional ethanol moiety here, as you can see here, and this led to what is called Capivasertib. It had a number for it. They always have numbers. It had a number first, then it was called Capivasertib, and now it has a commercial name, TrueCap. And as with the previous compounds, it's a potent inhibitor of AKT. But AstraZeneca minimized off-target effects, which was really good. And they showed that, as we had, that it showed pharmacodynamic activity and human tumor xenograft activity. And they did a lot of work on scheduling and are still doing quite a lot of work, as James has said. And it was in phase 3 clinical trial. I do need to change that bit because he told us it was approved. And he's right. It went into clinical development. The phase 1 was done. It was funded by AstraZeneca, and the lead investigator was at the Royal Marsden, which the ICR is associated with. But it was great for me because a lot of the biomarkers that we developed in my lab were actually we did contract research, basically, or contract analysis of the patient samples from the phase 1 clinical trial, which AstraZeneca paid for. And one of them was actually, and I'm looking at the time, I know I'm running over. But I have to tell you about this one assay because it was just so hilarious. It wasn't. It's always read scientific articles because you learn things. And there was a trial for something called for drugs called PARP inhibitors. And in that trial, they'd actually looked at a biomarker in human hair follicles. And I needed in phase 1 some form of biomarker that we could look at in dose escalation in the patients, because we're never going to get them to have mandatory biopsies during phase 1. These are patients that have failed every other therapy. And we thought, well, if we can do something less invasive that allows us to see whether we can see inhibition of AKT in some form of surrogate tissue, if it's not the tumor, and hair follicles are, because they're embedded in the epithelial layer. And so we looked in mouse hairs first when we were doing the in vivo studies and found that we could see inhibition of Pras40 phosphorylation, and then we tested it with ethical approval in our own hair follicles that we plucked ex vivo and then incubated with the compound, and eventually we initiated it in the clinical trial. And so we had to ask the patients in phase 1 whether they would be willing to have their eyebrow hairs plucked so that we could monitor phosphorylation of Pras40. But it was really important because we could see in the phase 1 of the single agent, as we increased the dose, more of the phosphorylation got inhibited. So we could see a direct tie between dosing, and this is what James said, and a biomarker modulation. And eventually this is some of the data from the phase 1. We also confirmed that in patients where we could see mutations in our pathway, that they were the best responders. Okay. And that was in the phase 1. It was very important. It's gone through phase clinical trial. Thank you, AstraZeneca, for putting it into 5 different phase 3 trials. That costs a huge amount of money. And we are so happy because it was approved last year. This is the clinician who led it. And one of the other people, because you have to recruit, someone mentioned this, it required international recruitment for thousands of patients for the phase 3. And this was the data from this trial for recruitment in the US, Europe, and the UK that led to this difference in median progression-free survival was enough to get approval with the FDA when combined with another drug in metastatic breast cancer, hormone receptor positive, so estrogen receptor positive. And this is the post from the FDA announcing it, which was on the 16th of November. And I found out through my old clinical fellow who is now and this is how time progresses, your people go on and do great things. He's now at MD Anderson Cancer Center as a professor doing clinical trials. He's a clinician with a PhD. And it was just great for me to hear it because he WhatsApped me because of the time difference and he found out first. And it's just, been a fantastic journey all the way from the start, from proposing it, and all the hundreds of probably thousands of people, including the patients. But what I've tried to explain to you is that, from proposing it as a drug target to CRUK, collaborating with Aztecs, and then, licensing our early compound to AstraZeneca, and all these different types of chemistry and biology has led to the identification of TrueCap, which is, yes, it's still in 5 phase 3s. It'll be approved, we hope, in other disease indications. And one of the things that I've been looking at in my lab is how tumours develop resistance to it, because tumours will eventually, and we need to be ready to know how to deal with that. And so I only just want to finish by saying that, science is a team activity, and I can't put everybody on here, but you sort of get the idea. And the final slide, which if you'll leave, I just want to, because it's really important, things I learned along the way, right? And this is all the way from my undergraduate research project to now. Take the initiative. If your supervisor can't help you with something, find someone who can. There is always somebody. Find a mentor, or even more. I still have one for something new that I'm doing. I don't need to explain it, but I found a mentor. And I also mentor people. So it's a two-way thing. Networking has been so important for me. All my jobs, it's really weird, were through networking. I'm not saying you're going to be able to do that, but finding out about jobs is through networking as well. Everyone fails at multiple points. It's just one of those things, and especially in science because of what we're doing. But just keep going, and be yourself. That's really important, and find a career that you enjoy, because you're going to be in it for a long time. And enjoy it, and also have a life. So this is the picture we had. There was a celebration at St. Paul's Cathedral, actually, for TrueCap, and this is Barry Davis from Aztecs, from AstraZeneca. It was funded by Aztecs, actually, but they invited all the AstraZeneca people. This was Ian, who's a lead chemist, and me. And this is us winning the AACR Team Site, the American Association of Cancer Research Team Science Award. And one of the contributing projects was the AKT inhibitor project. And this is just a reminder to, enjoy life. That's my daughter. And I'll stop there and take any questions.
Fantastic. Thank you so much, Michelle.
Right, I'll stop there.
We've already had one pop in. Hopefully we'll try and squeeze in perhaps one or two, because I'm sure there'll be lots of questions. So the first one's from Casey. So what have been your biggest challenges when collaborating with so many different people in different disciplines?
That's easy. Well, communicating with them is key. You have to keep people informed in different organizations. So, we had initially we had a project team that was between the ICR and Aztecs, and we had project meetings. But I think one of the key things is remain calm in the face of adversity. People are individuals and get upset. And the most important thing to do is listen to what they've got to say, even if they may have raised their voice, and remain calm. And also when people are giving you advice, even if you might not agree with it first, go away and think about it. I think it's remain calm and take time to think about things.
Yeah, I think that's a very good tip. Any other questions for Paul?
Whether there's anything else.
Any other questions? I think somebody's there. Any other questions at all?
You are welcome, Casey.
Perhaps if anyone has any more questions to pop into the chat, and we're short for time. But thank you again so much, Michelle, for joining us. It was brilliant to hear. It's always nice, isn't it, to be able to hear the full story, or 20 minutes of a full story.
Oh yeah. Somebody said, could you? Yeah, we could. Somebody did say it was how we did it. Actually, Tim looked during the mouse efficacy studies. We did some mice that had hairs, is the only way to explain it, and mouse with hairs, and we plucked their hair, a few of their hair follicles, and we could detect a loss of the phospho-threonine 246 signal. In fact, we published it. It's on, if you look at me and Tim Yap, I think it's in his first author paper. And so we validated it in what we call the preclinical setting before we went to the clinical setting. Oh, Wi-Fi 5. Okay, so I'll give my view. James may have another one. The thing is that I mean, it is a lot, but I think the thought is that it will, because of all the preclinical work that's been done with the final compound AstraZeneca, is clear, and also remember it's been in phase 2 in those disease indications as well. But the preclinical data from AstraZeneca indicates that it should have efficacy in both prostate and breast cancer. They're both hormone responsive, and also in different combinations of the AKT inhibitor with different drugs. And so that's why 5 phase 3 clinical trials.